right, welcome to Virology. I am Vincent Racaniello. I'm going to be your professor for this entire course. There's nobody else that teaches it but me. And hopefully I'm good. Otherwise you're in trouble. If you would like to understand life on Earth, if you want to understand human health and disease, you need to know about viruses. And that is what this course is all about, to give you everything you know to understand the interaction of viruses with the planet. I am a professor at the medical school. I've been there for 40 years doing research on viruses. And I think 12 years ago, I started to teach this class. Now, you have to understand that at a medical school, you don't have to teach. You have to teach medical students, but the clinicians do that. The basic scientists, such as myself, all we have to do is do research. We have to raise money to do our research. We don't have to teach. But I've always loved teaching. And I realized there was no virology course down here, which is a shame, right? You're paying a lot of money to go to school here, or maybe your parents are paying a lot of money, and you don't have a virology course, which in my opinion is the most important course on the planet. Most places don't even have a virology course. So I started teaching this 12 years ago. It started small, 30 students, then 40, 50, 60, 100. The most we ever had was 175 one year. That was a lot. And we have moved around to different classrooms. But I love doing this. I love viruses more than anything else on the planet. And don't tell my family that. I think they're amazing, and I want to impart to you some of my amazement for viruses. They're really incredible entities. And notice I use a word, entity. So what exactly are they? Well, we're going to define it today. We live in a cloud of viruses. Viruses infect every living thing on the planet. I was amazed to hear that people during the pandemic didn't know that everything was infected with viruses. They thought there were a few viruses around and now and then they give trouble, but for the most part, not. But in fact, every living thing on the planet has many viruses that infect it. There are viruses in the air in this room of different sorts. You are breathing them out and you are inhaling them. Toilets are full of viruses. Did you know that toilets generate aerosols when you flush them and that you inhale those aerosols? Man, you better close the lid when you flush the toilet, if there is a lid. I often see if there's a lid or not. Not much you can do if there isn't one. So viruses are everywhere. We regularly eat billions of virus particles. If you have coleslaw, it's full of viruses because the insects that crawl over the cabbage have viruses in them. So we take in and give off billions of virus particles on a regular basis. And in fact, our genomes have in it integrated nucleic acids of viruses. And some of those are actually expressed, and we'll talk a little bit about those today. And some of the genes of those viruses that are integrated in us have actually been repurposed by us to do other things. Really remarkable. So everything is infected. The number of viruses on the planet is simply staggering. This is the game of viruses, if you want to say that. They make a lot of progeny because the likelihood that any one virus particle will find a cell is very low. So they just spew out billions and billions of particles, and maybe a few make it. So here's an example of the numbers. In the waters of the planet Earth, mostly the oceans, there are over 10 to the 30th particles of bacteriophages. These are viruses that infect bacteria. 10 to the 30th. You can't comprehend that number, right? It's just too big. So here are some things to tell you what that means. So a single particle weighs about a femtogram. So if you multiply a femtogram times 10 to the 30, you come up with a biomass that exceeds that of elephants by 1,000-fold. Elephants, all the elephants on the planet, all the viruses weigh more than those by 1,000-fold. If you lined those viruses up head to tail, they would go 100 million light years into space. These are things you can't even see. That's how big 10 to the 30th is. Huge number, and that's just in the oceans. And you know what's amazing? We didn't even know this 15 years ago. We thought that the oceans mostly had whales and fishes and that sort of thing in it, but it turns out incredible microbial communities and viruses that infect them. Whales are infected with viruses. They're huge, right? So they excrete huge numbers of viruses. They're commonly infected with viruses called caliciviruses. This is a virus which is part of a family of viruses that infect us called noroviruses. If you've ever had vomiting and diarrhea for 2 days, a day or two, that's a calicivirus. It's a norovirus. We'll talk about those later. In whales, they can cause diseases, rashes, blisters, gastroenteritis. They can infect humans, in fact, and infected whales excrete over 10 to the 13 caliciviruses every day. So those go into the waters, of course, and they may infect other marine mammals as well. So the numbers are huge. I like to say that viruses are not just messengers of bad news. Most viruses on the planet are actually not bad. If you, by bad, mean cause disease. Most of the viruses don't cause any disease. Now, if you look in the oceans, you see that viruses have enormous roles to play in what we call biogeochemical cycling. That is the recycling of organic matter to form the elements which can be reused. We didn't realize this 15 years ago, but if you took all the viruses away, we'd have problems getting carbon and phosphorus and sulfur and nitrogen out of organic materials. In a liter of seawater, there are more virus particles than all the people on Earth, just a liter of seawater. And in a teaspoon, many millions of virus particles. So the next time you go to the ocean and you take water in your mouth and you spit it out because it's fun to do, you're aerosolizing lots and lots of viruses, but they're probably harmless for humans. Now on this chart here, on the left, we have biomass. So the mass of things in the ocean, they're prokaryotes rule, bacteria, right? And viruses and protists, a very small slice of that pie. But if you look at abundance, particle abundance, if you count particles, what you will find is that the viruses constitute 94% of all the particles in the ocean. So again, the numbers game. Viruses have huge numbers of offspring. And we will talk about this more in our ecology lecture at the end, but as I mentioned, they're important for biogeochemical cycling. They infect, say, bacteria, and the bacteria lyse. There's something like 10, is it 10 to the 30th? I forgot the number, but a lot of infections per second in the surface of the water, phages infecting bacteria, huge numbers. That liberates particulate organic matter, which sinks down into the lower depths. It's degraded further, and it's recycled into the elements. So really important constituents of that cycling. Other viruses are also present in huge numbers. HIV, human immunodeficiency virus. The only virus, by the way, which gets its own lecture in this course. This is not a virus by virus course, but HIV is so important. It is a pandemic still, right? There are 2 pandemics ongoing, and depending on how you define pandemic, there may be more. Pandemic is not really a virological definition, it's more of a sociological WHO one, but HIV, AIDS is a pandemic. There are 10 to the 16th genomes on the planet today. We know how many people are infected, we know how many virus particles each of them have, so huge numbers. We will talk a lot about HIV. So how infected are you? You have at least a dozen different herpes viruses. Every one of you, I would bet a lot of money that every one of you have at least a few herpes viruses, if not more. You get infected very young in life, from your parents mostly, because they're kissing you, and most of these viruses are passed through saliva. And they include herpes simplex viruses, varicella zoster, cytomegalovirus, Epstein-Barr, and then HHV 6, 7, and 8. We'll talk about these separately. But once you are infected, it is for life. You cannot get rid of these viruses. They remain in various parts of your body in a form we call latent. Periodically, they reactivate and you shed infectious virus. You may have a cold sore in the case of herpes, or you may have no symptoms. And that's how they spread from person to person. But you also have many other viruses in you besides these. These are the ones that are well studied. You know that we each have a microbiome in us, right? Every tissue and organ in us carries a different kind of bacteria, as shown here. Well, we also have a virome, which is less studied. It's less studied because it's hard to study. The microbiome is pretty easy because all you need to do is to take samples of each of these tissues and sequence the ribosomal RNA. And that will tell you what kind of bacteria is present. Ribosomal RNA is quite short, so it's easy to sequence. There's no ribosomal RNA in viruses, of course, so you have to sequence most of the viral genome, which is harder, and then you have to figure out what virus it is. But nevertheless, we know that in all of these places where there are unique bacteria, there are also unique viruses, not just bacteriophages. There certainly are bacteriophages in each of these compartments that infect the microbiome, but there are also viruses that infect our cells themselves. And this is a picture of that. There on the left-hand part of the slide is a diagram of a human, and the pie charts show you the numbers of DNA and RNA viruses in each compartment, nervous system, respiratory tract, etc. So viruses can have either DNA or RNA as genetic material. They're the only organism on Earth that has RNA. Everything else is DNA-based. And we will talk about why that is or how that came about. So we have both RNA and DNA viruses. Look, on your skin, on your hair and nails, you have viruses, even in your blood. And so all of us have this. Our blood supply is full of viruses. You cannot throw away blood just because it has viruses in it, because you would have no blood supply. And we haven't yet made artificial blood, so we can't do that. We throw away blood when it has a virus that's known to cause disease. But most of the other viruses we leave alone. So most of these viruses we think are benign. And we suspect that some of them may be helpful, actually, although that's harder to find out. Now, the other picture here on the right illustrates how we can sample the virome of different animals or hosts using different methods. And this is a sampling of whales off the coast of Australia. And at the bottom is a drone. And the drone has on it a big petri dish, which is going to open up as the whales exhale. And of course, there's a scientist on a boat nearby, and they're controlling the drone, and they're opening the lid when the whales blow. So let's see if the movie will play. There you go. See that? So let's do it again. The whales blow. He or she opens up the petri dish, closes. Now it's got liquid in it. They bring it to the lab and they can sequence it. And you can find in whale breath lots of different viruses. You have to say whale breath because you don't know what's actually in the whale. It may not be representative of what's in the whale. It might be a subset. So harder to get inside the whale, right? Only if the whale washes up dead, and then even then it may not be representative. So this is the best that we can do. We find many, many different viruses there. Lots of viruses in every organism, and of course every organism also has viral nucleic acid in the form of DNA integrated into their genome. This is a map of the human genome, 3.2 billion bases, with all the different elements that are encoded, which many of you may have encountered in some of your biology courses. But look at the protein coding genes, only 1.5% of our DNA. I find that amazing every year. And there are lots of other sequences, but the ones I want to point out here are the LTR retrotransposons. About 8% of our genome is viral. These are from viruses that have infected us and integrated their DNA into our genome. And in fact, some of these other elements, lines and signs, have also elements reminiscent of viruses, which we're going to talk about. So big parts of our genome are related to viruses. Now, of course, not all viruses are benign or beneficial. Many of them cause disease, not just in us, but in other animals. Dogs and cats get sick, minks, deer, all the hosts out there can get sick. And in humans, we are concerned about human disease, of course, and that drives a lot of virology. And this is a graph of the causes of 2017 global deaths. there's a very nice website, ourworldindata.org, has all kinds of data, but this year they did not have this same chart, which is arranged by disease. As you can see, the cardiovascular diseases are the biggest killer. So this is for 1 year, about 18 million deaths due to cardiovascular diseases. And then we have cancers. And of course, respiratory diseases, 3.91 million in 2017. But COVID, of course, has killed at least 6.7 million people globally as of a few days ago. I say at least because this is an underestimate. We often don't mark on the death certificate cause of death COVID. Could be heart attack or stroke or something else secondary to COVID, but often COVID isn't mentioned. So this is an underestimate. So that would put it in the number 3 position here. So there's many respiratory diseases, of course, are caused by viruses. So I have the arrow there to show that. Then we have diarrheal diseases. Many are caused by viruses, bacteria as well. HIV/AIDS, 1 million people died in 2017. And that's a pandemic that's been going since 1981. A lot of people still dying. And then hepatitis down there at the bottom. A number of viruses cause hepatitis. So don't underestimate the role of viruses in human disease, despite the fact that many of them may be benign. So here's a final summary for you of the COVID pandemic. These are graphs of, on the upper left, daily new cases. This is a 7-day running average, starting with January 2020 when the pandemic began. By the way, I was teaching this course in January 2020. On the first day I said, anybody hear this respiratory outbreak in Wuhan? And 3 people raised their hand. That was, January 17th or 18th. And every class after that, for the first 5 minutes, I would put up the Johns Hopkins chart and we would see the dots increasing in number. And then they started to spread outside China. And then, of course, spring break, that was it. We all went home and never came back. And I'm freaking out because I'm teaching a virology course and it's making me stop teaching. That was just the weirdest thing I've ever encountered. So far, you can see there have been periodic waves of infections for various reasons. New variants have arisen, seasonal, people getting together. We lock down, then we come out of lockdown, boom, you have a lot of cases. People go back to school, they go on vacation. And that's on the upper left, the number of cases, 671 million cases. But again, an underestimate because we don't test everyone. I don't know how much of an underestimate. And then the deaths on the bottom right there. Daily deaths, you can see, again, many waves. And lately it's been much lower because of vaccination. But it's not zero, and we can talk about reasons for that later on. So despite this, in these diseases I've just shown you, most of the viruses that infect us don't really cause disease. They seem to not have an impact on our health and well-being. So why is that? Why are we able to handle most virus infections? Well, many viruses we eat and they pass through us. They're viruses of insects or plants and they can't infect us. And so here's an interesting study where they got cabbage from 5 different supermarkets in Washington, D.C., and they looked for viruses and they found that each serving, about 100 square centimeters of cabbage, had up to 10 to the 8th particles of an insect virus. And the insect is the cabbage looper, shown there on the right in green. It looks like a caterpillar, right? This virus infects them. They crawl over the cabbage and they shed virus, and you can't wash it off. It adheres very, very tightly. So you eat it, and it passes through you. It's an insect virus. That's the virus, that long virus particle shown there. That's the virus, and it will not infect human cells. So they pass through us. Now, if you take human feces, there's actually a company where you can send your feces to, and they will sequence it and tell you what viruses you have in your poop. I get emails all the time from people freaked out. Oh my God, I have a pepper mild model virus in my poop. What's that? Is that a problem? Well, pepper mild model virus is a virus that infects peppers. It's a RNA virus. It's filamentous, as you can see at the bottom, and the pepper on the right is infected. So it gets this mottled surface on the pepper. And if you sequence your poop, 91% of the RNA you find is this virus, I guess people like peppers. And also ground pepper as well can have this in it. So many viruses pass through us, and these are just two examples. Some viruses are beneficial. We have lots of examples of viruses that are beneficial for non-humans. It's hard to do these experiments in humans. So here are two examples. On the left, that's a grass, Dicanthelium lanuginosum. It grows near the hot springs. If you ever go to Yellowstone and see the pools of hot water, there are grasses that grow around it. And these are heat-resistant grasses. And the reason they're heat-resistant is because they have in them a fungus that's infected by a virus. And you can bring these grasses into the lab and grow them at high temperatures, 55 degrees centigrade. You can take out the fungus and the virus, and the plants will die. If you just put in the fungus without the virus, the plant will also die. So you need the fungus-virus combination to somehow confer thermotolerance. And this is a theme, actually, in many wild plants. If you go into the forest and take a plant, they often are thermotolerant, drought-tolerant, tolerant of many things, because they have viruses in them. And the crops that we use for our food, for corn and wheat and potatoes, they're what we call monocultures. They're derived from a single seed, and they have no viruses in them. So they're kind of wimpy. And so one thing that people are trying to do is to introduce viruses to make them more robust. So that's one example. And then on the right is a virus that is good for wasps, but not so much for caterpillars. So that's a parasitoid wasp. And what it does is it injects its eggs into a caterpillar. And you see it's got a long injector in its tail, and the eggs are shown in as oval in yellow. So the wasp egg goes in, and along with the egg goes virus particles. Those blue particles are called pallidnaviruses. They're encoded in the wasp genome. So as the egg is produced, the viruses are produced, and they both go into the caterpillar together. And what those viruses do is they immunosuppress the caterpillar, because otherwise, without the virus, the caterpillar rejects the wasp egg, and that's the end of it. Isn't that amazing? The viruses express genes that immunosuppress the caterpillar and prevent it from rejecting the wasp egg. So the wasp egg grows, it becomes a little wasp, and it eats its way out. So this is the original alien, right? It eats its way out of the caterpillar, not the movie. But anyway, the virus is obviously good for the wasp, and it has evolved over many years to be part of the wasp genome. Really interesting. It doesn't do anything to the wasp. Now, we do have something close to humans, a mammal. This happens to be a mouse experiment, and you can argue whether it's relevant or not to humans, but I'll tell you the result. This shows us that a virus that infects the enteric tract of mice is beneficial. So in mice, these are sections of small intestine. Conventional mice here on the right, very nice villi, very nicely formed. And there's an immune system in the gut, which consists of T cells. And so those are staining brown in the lower two segments. Those brown cells are T cells. If you grow mice under germ-free conditions, where they have limited numbers of bacteria, that prevents the gut from developing properly. So you can see on the left, GF is germ-free. The villi are malformed, and underneath there are no lymphocytes. So somehow the bacteria in the mouse gut, the microbiome, facilitates the development of the villi and of the immune system. Now, if you take these germ-free mice and now infect them with a murine norovirus. So norovirus is a calicivirus related to that same virus that infected whales that we saw some time ago. You partially restore the defect. So the villi look a little better. It's not 100%, but there are some lymphocytes in the villi as well. So you have a partial restoration. The idea being that somehow norovirus, which colonizes mice, contributes to the formation of the gut and the gut immune system. So you can't do these experiments in people. And so it's hard to know if the same would happen in people. We're going to have to wait for other experiments. And one year in this class, someone came up afterwards and said, if we had broad spectrum antivirals, you could find out, right? And yes, someday we're going to have broad spectrum antivirals. We're going to treat people and get rid of all of their viruses because we're trying to get rid of a few. And then we'll see if that's a problem. we're going to inadvertently see. Because as you know, we treat people with antibiotics and we cause problems because we cause imbalances in the microbiome. So it may be that we'll see similar things when we have a broad spectrum antiviral. We don't have one yet. Now, the other reason most viruses don't cause any problems in us is because we have this amazing immune system, which blows me away on a daily basis. The more I learn about it, it's just amazing. And it keeps most virus infections in check. We will devote 2 lectures to this, which is just a drop in the bucket. I know many of you have taken the immunology course here, so you know a little more, but we'll talk about how viruses interact with the immune system. But the immune system keeps our virome in check. And we know this because if your immune system is down, why would it be down? Well, if you have an organ transplant and you have to take immunosuppressive drugs, then viruses begin to multiply and cause problems. A whole field of transplantation virology has arisen to treat patients who are getting liver transplants to make sure the viruses in them don't take over and kill them. Or you could have a virus infection that immunosuppresses you, like HIV is strongly immunosuppressive. Other viruses will then arise and cause problems. And other viruses will immunosuppress as well. Measles virus immunosuppresses you. So we depend on this wonderful balance between the immune system and viruses to keep us healthy. Now, not all, as you may gather, I have said multiple times already, not all viruses make you sick. And I wanted to give you this wonderful example of a DNA virus called polyomavirus that infects everyone. You get infected early in life, and you typically get the virus from your family members. And so it turns out that this is a wonderful virus to track human migration because you get slightly different viruses of this polyomavirus type depending on where you live. And so this map shows you migration of humans out of Africa, where we began as Homo sapiens. And the dotted purple line is the migration based on the genome sequencing of humans, right? Out of Africa into Europe, eventually Asia and North America. But the black line is the migration based on polyomavirus. We can take blood from various people and see what kind of polyomavirus infection they had. We can trace migration, and you can see it follows the genome, but it's even more detailed. You can see movement into Australasia and South America as well. So it's a virus that doesn't make us sick, but it can give us interesting information. Now, viruses also shape populations. Obviously, if the viruses are killing cells or animals, they can do that. And a great example, which we'll talk about more, are viruses that infect eukaryotic phytoplankton. So this is a bloom. This is an algal bloom in the ocean. It's a satellite photograph. Under certain conditions, they get huge, and you can see them from outer space. It's an organism called Emiliana huxleyi. And so when the sun comes out, the blooms increase, and then there are always some viruses around that will infect this organism. The viruses collapse the bloom, and it goes away. So the viruses are shaping these populations. And if you've seen green or blue or brown algal blooms in various waters, they're usually collapsed by virus infections. So this is a little introduction to why I think viruses are amazing, and we're going to explore all of these points in this course. But maybe the most important thing to say at the onset is that virology is an integrative science. It is not just one science. It's not chemistry, and I don't mean to denigrate chemistry. Chemistry is wonderful, but it's chemistry. And then there's biochemistry and there's other sciences, but virology takes all of them to understand how viruses work. You need to know cell biology, biochemistry, organic chemistry. You need to know sociology to see how people interact with each other. Psychology. All sorts of fields need to be understood in order to understand virology. And that's what I mean by it being an integrated science. Now, we're going to talk mainly about the viruses themselves in this course, but you'll get a sense for how you would need to know other things to understand them. So my goals for this course are to give you the big picture of virology. And I want you to think about it as an integrative science, not a collection of viruses. I don't want to teach you about influenza and herpes and polio. You will learn about some viruses, but a virus course is a separate course, and Maybe one day I will teach that, but that's not what this is. I want you to get the principles here. You're going to learn fundamentals. And you know, people are afraid of things that they don't understand. So all the misinformation in our society, and relating to viruses, that would be misinformation about vaccines, about mask use, about all sorts of issues surrounding COVID. All the misinformation comes from people being scared. They don't know where to get their information, and they turn to the wrong source. But you're going to get the fundamentals here. You're going to be able to understand and no longer be afraid, because you won't be uninformed. And hopefully you can pass the information on to others and help inform them about it as well. Now, one of my pet peeves that you will hear me, I do some ranting in this course. I figure it's my pulpit and I can rant, but I have good based rants, and one of them is all about mutation. We're going to talk a lot about this in the course. And the COVID pandemic has given me lots of raw material to complain about. So let me just give you a little example. These are various headlines from the very beginning of the pandemic, January 2020, December, and it's all about mutation. Coronavirus could mutate. China warns virus could mutate. There is the possibility of viral mutation. The coronavirus is mutating. These are like headlines saying the earth is round. These are the dumbest headlines you could write, because viruses are always mutating. Every time a virus multiplies, you get mutations. So these headlines, and you know, when you talk to the authors of these articles, they say, oh, but the headline writers did it. I didn't do it. They always pass the buck to the headline writers. Well, maybe some of you will be headline writers someday, and so you will know what kind of headlines not to make. And I'm not explaining this right now. I'm going to go into it later, why it makes no sense to write that viruses are mutating. There's something else that you should be saying. What is a virus anyway? So here we are, 40 minutes in. What's a virus? So this is my definition. And by the way, you guys know ChatGPT, right? Is that what it's called, ChatGPT? Yeah. So I asked it, what is a virus? And it gave a pretty good definition, actually. It was too long, so mine is better because it's short. An infectious, obligate intracellular parasite with genetic material, can be DNA or RNA, got a protein coat and sometimes a membrane. So ChatGPT didn't have some of these complicated words. It didn't say infectious, obligate intracellular parasite. So what does that mean? Infectious, of course, means the virus can enter a cell or a host and reproduce and then spread to another one. Obligate intracellular parasite means the virus has to get inside of a cell in order to reproduce. And being a parasite means you take something from the host. You take something away from the host. So viruses take all sorts of materials in order to reproduce themselves. Sometimes they kill the host, which is the ultimate parasite, right? But you can have different levels of parasitism. Then we can have DNA or RNA. You have to have at minimum a protein coat. So some of these viruses here have just a protein coat. So here on the lower left is poliovirus, which just has a protein coat surrounding an RNA genome. This is adenovirus with the little Sputnik-looking things. There's DNA inside of that protein coat. But many viruses also have a membrane around that as well. So here's a coronavirus where we have a membrane around the nucleic acid, which has got protein on it as well. And many of these other viruses, which we'll encounter in this course, have membranes as well. So this is a one-sentence definition of what is a virus. So the ChatGPT was like 3 paragraphs. A little too verbose, as you get lost. Because viruses are obligate intracellular parasites or molecular parasites, then studying them tells you a lot about the host. And we've discovered many host functions by studying viruses. Like splicing of mRNA was discovered in a virus-infected cell, and many others as well. And so whatever host you study, whether it's a mosquito or a eukaryotic flagellate or humans or plants, you can learn something about the host, which is a nice benefit. And now the question comes, are viruses alive? Are they living? So for a while I had a poll on my blog. It disappeared, but after so many, 6,000 or so answers, this is what we got. 2,000 voted yes, 2,200 no, and then 2,000 something in between. I mean, I put these answers in myself. I don't think I should have put something in between, and I don't know what's in between, right? So, but it was a long time ago. And then a few people don't know. So are they alive or not? So I thought about this a long time, and here's my take on it. You don't have to agree. I think a virus is an organism with two phases. The one phase is the particle. So on the left here is a polyomavirus. That's the virus particle. So when I say particle, I mean the virus itself. So some people are confused by me using the word particle, but what I mean is the intact virus. Infectious particle, all right, that is not living. It can't do anything. If I had a tube of virus here, it would just sit there, it would never reproduce, it would never evolve, it would never mutate, because it needs a cell. So how can you say a virus is living if you're referring to the virus particle? And most people, when they say virus, they usually refer to that. However, when that virus infects a cell, it transforms the cell into a virus-making factory. And the infected cell is certainly alive. It's the other phase of a virus. It's the living infected cell. So that's why I say two phases. The virus particle, not living. The infected cell is living. It's the embodiment of the virus because it's making more viruses. The virus has completely reprogrammed the cell to make more viruses. So it's certainly part of this organism called the virus. So that's what I think, and makes it perfect sense, but I think it's very difficult for many people to grasp, and still, even my colleagues, my science colleagues, talk about viruses being alive. And if you mean the particle, that cannot be. Now, I want to warn you that when we talk about viruses, don't make them human, please. Don't anthropomorphize them, right? They don't think. If you read popular science articles, they often do this to make it easier for you to grasp. A virus's goal is to do this. No, a virus doesn't have any goal. It's not alive. It is a chemical that is reacting to its environment. You can't say viruses think, employ, ensure, exhibit, display, because they're passive agents. Now, why do I say this? Am I just being a pain in the butt? No, there's a reason. There's a reason for this, is because if you start giving human qualities to a virus, you're going to get it wrong at some point, because you have no idea what is driving the virus. You're putting human qualities on it, and that is almost certain to be wrong. So you could say the virus mutated because it wanted to do this, but you'd be making an assumption that isn't tested. So that's why I say don't give viruses human qualities. It'll cloud your understanding of their evolution. There are better ways to express what's happening. If we would like to know why a certain mutation is selected for, we talk about it in those terms. We don't say a virus wanted to do this. We don't say a virus wants to transmit to a new host. There simply is no desire on the part of a virus. It is passively moving from host to host. If it happens to encounter the right host, it'll multiply and move on. If it doesn't, it becomes extinct. And certainly many viruses have done that over time. All right, try and be aware of that. It's really important. Now, viruses are small. Part of the original definition I used to give in this course is viruses are very small. So here's a slide that kind of illustrates that. We have E. coli, 100,000x, and there's a bacteriophage attached to the E. coli. And then above the E. coli is a rod-shaped virus. That's tobacco mosaic virus, the first virus discovered. That's C. And then above that in D is HIV-1. So you can see they're smaller than E. coli. And then that little panel in A, we magnify that 1,000,000 times, and then on the right you can see another virus. H is actually poliovirus, and G is a ribosome. So poliovirus is about the size of a ribosome. And then above it we have F is an antibody, C is a tRNA, I believe, and these are all various molecules inside of a cell. So they can be quite small. However, they are actually bigger than we thought they were. Here's a nice scale to give you a sense of this. So here we have arranged from plant cells, and this is the size here, animal cells, bacteria, viruses, ribosomes, proteins, small molecules, and atoms. So viruses somewhere in the middle here. And the various techniques that you need to see viruses. You can see, most viruses you can't see in an electron microscope. You need in a light microscope, you need an electron microscope, or you can do cryo-EM or X-ray crystallography, things that we're going to talk about later. And here on the bottom is a picture of a cell to give you, again, more sense of scale. Here's a cell with its nucleus. And there on the surface are some virus particles, and they include poliovirus, again, about the size of ribosomes. Ribosomes are 20 nanometers, polio about 30. And there's the herpes virus, which is substantially bigger. So in the early days of this course, I used to say a virus is a small intracellular obligate parasite. But then we discovered giant viruses. So if you're interested in how many viruses can fit on the head of a pin, by the way, there's a website from which I took this. There's the head of a pin. That is a hair laid on top of it. There's a dust mite. And in this boxed area are a variety of organisms. We have red blood cells, we have bacteria, yeast, pollen. And in there are some viruses. That's actually Ebola virus, which is quite large. So if it were rhinovirus, which is about the size of polio, you could put 500 million rhinoviruses on the head of this pin, huge numbers. And when you sneeze, you are firing out droplets that contain many, many different viruses that can infect many people. Now, about 15 years ago, we discovered really big viruses, bigger than any we had seen before. And those are the Mimi viruses shown here. And in comparison, we have rhinovirus, HIV, which was considered quite large. Here's a Mimi virus, which was even bigger. And here's a cell infected with Mimi viruses, which I think is kind of cute. Looks like it's looking at you. Subsequently, we found even bigger viruses. And now we have a whole collection of viruses that we call giant viruses, where the particles can be seen in a light microscope. This is Pandoravirus, and these are Pandoravirus particles photographed in a light microscope. They're 1.5 microns in length. So very few viruses are visible in the light microscope. This is one of them. Why is it called Pandoravirus? Well, when the genome was sequenced, which was about 2.5 million base pairs of double-stranded DNA, 90% of the genes didn't look like anything we'd ever seen. No protein looked like any of them, so they called it Pandoravirus because we didn't know what was inside the box. So we now have very, very big viruses. Now, the way viruses reproduce is important to understand. They're very different from our cells and from bacteria. Viruses reproduce by making the parts and assembling them in cells. They don't reproduce by binary fission. So binary fission is when you take a single cell, like this single bacterium, you put it in broth, and it doubles. It becomes 2 and 4 and 8 and 16 and so forth. That's binary division. And so the number of cells increases logarithmically with time. But cells don't do that. They don't divide. They go into a cell, they make parts to make new virus particles, and then the new virus particles are assembled. And so here is an experiment where we infect cells, and then we ask, is there infectious virus present at different times after infection? And you can see there's a period of time when we don't see any viruses. That's because the nucleic acid comes out of the viruses that we put into the cells, and it directs the synthesis of proteins. And eventually those proteins assemble into new virus particles, and then we can see new infectious particles being made. So this eclipse period is key to distinguishing viruses from bacteria, because there's no eclipse when we put bacteria into broth. They begin to divide relatively quickly. So an essential difference between viruses and bacteria. Okay, how old are viruses? They're very old. We will learn in a different lecture how you can date viruses. But the best estimate so far we have is that some of them arose in the sea about 450 million years ago. That would put them in the Ordovician period there, where the first land plants were appearing. And those are the kinds of organisms that lived in the oceans at that time. But in fact, I will tell you when we talk about evolution that viruses probably existed before cells arose on the planet. And I'm sure you're thinking, wait a minute, you just said viruses were obligate intracellular parasites. So what are you talking about? Well, we think before cells, right, there were kind of an organic soup forming on the planet. And we believe that self-replicating nucleic acids arose that could propagate on their own. It was probably RNA, and those were the precursors of viruses. And we will talk about that later on. And that's billions of years ago when the planet cooled and it was possible for these chemicals to form. More recently, though, we see references in the historical literature to viruses, that vase there, or from 700 BCE, talks about a rabid Hector, which rabies, of course, caused by a virus. And this carving on the right from 1500 BCE is an Egyptian priest with a withered leg that looks like he had polio. So we can find lots of references to what look like viral diseases. Of course, no one knew what viruses were at these times. No one knew what infections were. We did start to vaccinate, though, in the 11th century. In China, they realized that people who recovered from certain diseases never got them again. So they said, let's give people the diseases on purpose. So that was called variolation. It was done with smallpox. And it made its way to Europe. And Lady Montagu was the British ambassador to Turkey. And she saw this being done in Turkey, so she brought it back to the UK. We had no idea what was going on. We just knew that people who got some kind of disease never got it again. So in the case of smallpox, they would take the pustules and you blow them into the nose of someone, you can see there, or rub them on the skin. And unfortunately, that had a 10 to 20% fatality rate, which would not be approved by the FDA these days. But they thought it was better than taking your chance. Now, in the 1790s, Edward Jenner in the UK did experiments to establish vaccination, and we're going to talk about those when we talk about vaccines. But again, Jenner had no idea what was going on. We didn't know anything about any kind of infectious agent, no bacteria, no viruses at the time. The concept of microorganisms took some time to be established. We had Leeuwenhoek in the 1700s who made the microscope, the first microscope. And that's important because people up until then thought everything they saw was all that there was on Earth. There's nothing smaller. And he showed that there were tiny things that existed in fluids, including bacteria. Pasteur, in the middle there, found that bacteria could do various things. They could ferment wine and cheese. And then Edward Koch in Germany found that some bacteria could cause disease. So this established this concept that bacteria, at least, could cause disease and even had beneficial properties as well. So then where does virus come in? If you take a look at the historical literature, you can find virus as early as 1728, and it's used in a different way from what we'd use it today. It was used to describe something that causes an infectious disease. So by this time, it's recognized that people give diseases to one another, and they were called infectious diseases. And they were called viruses. Virus means liquid or poison in Latin. So viruses were thought to be liquids and passed from one person to another. And in fact, Pasteur said famously that every virus is a microbe. But he wasn't talking about the viruses that we are going to talk about in this course. He was talking about anything that made you sick, any infectious agent. And he knew about bacteria at that point. So he was saying basically bacteria or microbes, or viruses. A key event in our understanding of viruses came from this guy Chamberland. He used to work for Pasteur in Paris, and he found that the water that he used for his experiments. The water coming out of the tap was really dirty, so he wanted to clean it up. So he made this filter. It was a porcelain filter, and he used it to filter the water to remove bacteria to use in his experiments. And Pasteur was doing experiments on rabies. We knew that dogs got rabies, and he found that whatever was causing rabies in dogs was passing through these filters. So he said, oh, it's a small bacterium causing rabies, right? Okay. Then at the end of the 1800s, by then tobacco is a big deal. People are smoking cigarettes and pipes and cigars and so forth. Huge industry. And tobacco develops this disease called tobacco mosaic disease, where the leaves have this mottled look to them. And so scientists want to figure out what it is and maybe get rid of it. And so they do some experiments, and they use this filter of Chamberland. They grind up the leaves in a buffer, and they ask, does the agent go through, or is it retained on the filter? If it's retained, it would be a big bacterium. So two different groups did this, Ivanovsky and Bayerink, and they found that the agent passes through filters that retain bacteria. So some people thought, well, it's a small bacterium. But others said, oh, it's something different, because it doesn't multiply in broth. So the agent goes through this filter. Today we have similar filters which you can buy. They have about a 0.2 micron pore size. It's roughly what the porcelain filter was. Viruses will go through them. Bacteria are retained. So the tobacco mosaic agent is going through the filter. And the key was it didn't multiply if you just put it in broth. It would only multiply if you put it on leaves. And that was the difference between it and bacteria. In 1898, the foot and mouth disease agent was shown to be filterable. Foot and mouth disease is a virus that causes disease in cattle. You can see mouth lesions here. This is agriculturally very important. You don't want this in your cattle farm if you're selling cows. That agent goes through the filter. And the key, again, is not just small, but it doesn't reproduce. If you put this agent in broth, it doesn't multiply. You have to put it in the cow or in the leaf, the tobacco leaf. So we have something small going through a filter. It doesn't reproduce except in the host. And this is used as an operational definition for viruses for some time. They're called filterable viruses because some people still think they're small bacteria. So they call them filterable. They just go through the filter, so they're filterable viruses. And this is a list of discovery of some of these viruses. You can see all viruses causing human diseases: yellow fever, rabies, smallpox is variola, a poliovirus, 2 viruses of chickens in 1908 and 11, which we'll come back to, bacteriophages, influenza virus. Now, after 1933, something changes. This, by the way, is the timeline of discovery of bacteria. So as soon as Koch developed bacteriological methods, explosion in their discovery of tobacco mosaic virus in red, lots of viruses are discovered over the years. But in 1939, a key discovery was made by the electron microscope. So Helmut Ruska had made the first electron microscope in Germany in 1933, and the first electron micrographs of bacteria were made. This is one of the first ones. And lo and behold, they're particles. They're not liquids. This blew people away. And they're not bacteria either. This is the bacteria here. They're obviously something much smaller. So not until 1939 do we stop calling them filterable viruses. Now they're their own thing. They're just viruses. And then in this experiment, which we've already encountered, proved that they're not simply small bacteria. So bacteria, you put them in a broth, they begin to divide right away. Viruses, you infect the host, there's an eclipse period because the viruses need to get in, start making the parts, and assemble new virus particles. So now we just call them viruses. They're not filterable viruses. Now we have pictures of lots of viruses showing their particulate: bacteriophage, tobacco mosaic, rabies, poliovirus. It's quite clear that they're different from bacteria. We can solve structures now of viruses. This is polioviruses at 2 different resolutions: very high resolution, where you can see all the atoms, and a much lower resolution where you can only see the surface. And we'll talk about how these are done in a subsequent lecture. And we now classify viruses because we discover so many of them. And we typically just nowadays just sequence the genome and we can immediately classify it into all the existing viruses that we had. In the old days, we used to use other criteria like the kind of shell, whether it had a lipid, the dimensions, but we don't use any of them. SARS-CoV-2, first sequenced, immediately it's a coronavirus. That's it. You don't have to do anything else, and we'll talk a little bit about that as well. We use the Linnaean system for classifying, and for viruses, we typically talk about families, so Filoviridae, we talk about genera, that would be Ebola virus, and then species, Zaire or Reston or Sudan Ebola virus. These would be species. As I told you at the beginning of the 1900s, virus discovery is driven by disease. Now we just want to see what's out there. And we get samples and we sequence and we look for viruses. So here's a cool study done in China a number of years ago where they got 220 invertebrates, insects, crustacea, nematodes, etc. They ground them up and they did the sequence. We can do very, very rapid genome sequencing nowadays, and we can discover many, many new viruses this way. In these 220 invertebrates, they found 1,400 new viruses that we hadn't seen before. So you can do this with soil, you can do it with water, with your blood. It's just amazing what we can discover. So why do we care about any of this? Well, viruses outnumber cellular life by 10 to 1 on Earth. They're the greatest biodiversity on the planet. They drive biogeochemical cycles, as I told you. They shape host populations. They're likely to be beneficial. And of course, all those viruses out there are potential sources of new pathogens. So this sounds complicated, but in fact, as we move into this course, there are two facts that are going to help sort all this out. First of all viruses are parasites. They need to get into a cell in order to replicate. And all viruses need to make mRNA that can be translated by the host cell. No virus encodes a translation system, so the mRNA that viruses need to be translated by the host cell. So we'll see over the next couple of sessions how you can use that to simplify all those billions and billions of viruses out there. So next time we're going to talk about the infectious cycle. That is, what happens when a virus gets into a cell and then new virus particles are made.